Student, in this lecture we will see the remaining part of the translation. So in previous lecture we saw there the first step of the translation, that is the initiation of polypeptide chain. So remaining two steps, that is elongation of polypeptide chain and termination of polypeptide chain, that part we will see in today's lecture. Okay. During the elongation of polypeptide chain, that means there is a continuous addition of the amino acids in that polypeptide chain. It is called as a elongation of the polypeptide chain. Okay. So see student, that elongation of polypeptide chain means the addition of amino acid in that the polypeptide chain. So you have to remember the addition of amino acid in the polypeptide chain, it is takes place through the three steps. It is takes place through the three steps. What are those? The first step, it is the recognition of the codon. Okay, means the formation of the complementary codon anti-codon pairing. Okay, recognition of codon. Then the second step, it is most important, that is formation of the peptide bond. So this is the second step, and there the ribozyme enzyme, they plays a most important role. And the third most important step is the translocation. Okay, translocation means the movement of the ribosomal units over the mRNA. Okay, that means the movement it is takes place in the direction of the 5' into 3', and at each step the ribosome they move the length of one codon only. So student, let's see, these are the three steps. Okay, these three steps, how it is takes place during the elongation. Okay, so we have seen in previous lecture this initiation step. Okay, so this is the mRNA, just we have mentioned this is the mRNA. Okay, and this mRNA, they are having the 5' end and they are having the, this is the 3' end. But here the 5' end, just after that, here this region and this region, it is exposed. Okay, this region is exposed, no doubt. This region, it is called as a UTR region, means untranslated region. So that region it is not undergo translation. Okay, and before this region, means here it shows the attachment of the cap, that is the methyl guanosine triphosphate. Okay, and here just after that the untranslated region, we observed the poly-A tail. Okay, poly-A tail. We have seen in previous lecture. Okay, at P site, the first aminosyl tRNA complex, means charged tRNA molecule, they carry the activated amino acid. In case of eukaryote, they carry the methionine, and in case of the prokaryote, they carry the formyl methionine amino acid. So instead of saying the particular amino acid or presenting the particular amino acid, to that amino acid we represent that amino acid by using the amino acid AA1, means the first amino acid it is received, to that we call here for our convenience it is AA1, then second amino acid, to that we call AA2, like this way. Okay, so toward the first codon, the aminosyl tRNA complex, they directly receive that complex toward the P side, and that complex they form the complementary base pairing with the first codon of mRNA. Okay, in the sense what will be the anticodon of that complex? Here it is A, here it is U, UA, and GC. So the tRNA, they are having the anticodon UAC. That tRNA, they carry the first amino acid. In case of the eukaryote, they carry the methionine, and in case of the prokaryote, they carry the N-formyl methionine or formyl methionine. To that methionine or formyl methionine, we use the letter AA1. Okay. Then here you observe that A site, it is vacant site. Okay. A site, it is a vacant site. Then to that vacant site, the another aminoacyl tRNA complex means we have to say the another aminoacyl tRNA complex, they carry the amino acid and they are having the anticodon complementary to the second codon of mRNA. So that amino acid should be AA2. Okay, so this complex first they form the complementary base pairing with the second codon of mRNA. In this sense, the recognition of the codon of mRNA, this is the first step during the addition of amino acid. Okay, so they recognize its own complementary codon over the mRNA, and that complex they carry the amino acid towards the second codon, and that amino acid it is the AA2. Okay, AA2. आणि मग याचा अँटीकोडॉन हा या GCG याला कसा असेल, कॉम्प्लिमेंटरी असेल. म्हणजे इथं जर G असेल तर इथे C असेल, इथं C आहे म्हणजे इथं G असेल, इथं G आहे इथे C असेल. Okay, so you have to remember. So this is the first step, the recognition of codon. Okay, recognition of codon and the acceptance of the respective aminoacyl tRNA complex, in the sense charged tRNA molecule. Okay, then after receiving the two complexes, means first at the P side and it is followed by at the A side, then what happens, student? Immediately in the presence of ribozyme enzyme, okay, in the presence of the enzyme ribozyme, okay, in the presence of the enzyme ribozyme, the peptide bond it is developed in between the amino acid AA1 and AA2. So this is the peptide bond. So we have seen in chapter biomolecule in 11th standard how that peptide bond is formed. Okay, how that means amino group and carboxylic group they take part in the formation of the peptide bond. Okay, so this is the peptide bond. So the peptide bond it is formed. Okay, peptide bond it is formed. Then what happened? This tRNA they dislocate from its position. Okay, dislocate from its position because they carried the amino acid AA1. That amino acid they are attached with this second amino acid AA2 with the help of this peptide bond. Okay, with the help of this peptide bond. Then the next step. Okay, so this is the next step, that is the formation of peptide bond in the presence of the ribozyme enzyme. Then what is the third step? The third step it is the translocation. Remember, student, at each step the ribosome they move all along the length of mRNA, and at each step they move the length of one codon only. म्हणजे हा रायबोसोम हा mRNA वरती पुढे सरकतो, पण कुठल्या दिशेने पुढे सरकतो? दे मूव फ्रॉम 5 प्राइम एंड टुवर्ड दी 3 प्राइम एंड. आणि प्रत्येक वेळेस तो किती स्टेपने पुढे सरकतो, तर तो एका कोडॉनने पुढे सरकतो. तो तुम्हाला ह्या सेकंड डायग्राममध्ये इथं दिसतंय की हा AUG हा जो इनिशिएशन कोडॉन आहे तो बाहेर आलेला आहे, आणि त्याने पुढचा एक कोडॉन म्हणजे CCA, म्हणजे इथं दिसतोय तो CCA हा कोडॉन ॲक्वायर केला आहे. कारण रायबोसोम जो आहे तो आपण याच्या अगोदरच्या लेक्चरला इनिशिएशन ऑफ पॉलिपेप्टाइड चेनमध्ये बघितला की तो एका वेळेस किती कोडॉनला अटॅच होतो, फक्त 2 कोडॉनला अटॅच होतो. म्हणजे हा जो दुसरा कोडॉन आहे GCG आणि त्याच्यानंतरचा तिसरा जो आहे CCA, हे 2 कोडॉन हे रायबोसोमल सबयुनिटने ॲक्वायर केलेले आहेत. आणि पहिला जो कोडॉन आहे तो 5 प्राईम एंडच्या साईडला एक्सपोज झालेला आहे. ओके, सो यू हॅव टू रिमेंबर दिस वन. वेन व्हॉट हॅपन स्टुडंट, वेन द रायबोसोम दे मूव्ह फर्दर at each step they move the length of one codon. At that time what happened, due to formation of this peptide bond, the dipeptide it is formed at the A site. Okay, A site of the larger subunit of ribosome, the dipeptide is formed. म्हणजे ह्या जो सेकंड अमिनो ॲसिड टीआरएनए कॉम्प्लेक्स आहे त्याला पहिलं अमिनो ॲसिड अटॅच होतं. त्याच्यामुळं हा जो डायपेप्टाइड म्हणजे दोन अमिनो ॲसिड कनेक्ट झालेले पेप्टाइड बॉन्डने कनेक्ट केलेले आहेत, तो डायपेप्टाइड स्ट्रक्चर हे कोणत्या साईटला आपल्याला दिसतं तर ते ए साईटला दिसतं. बरोबर आहे. बट ड्युरिंग द मूवमेंट what happened, this dipeptide they move from the A side to P side. बघा ह्या फिगरमध्ये हे डायपेप्टाइड हे तुम्हाला ही जी ए साईट आहे त्या आणि ही दुसरी कुठली साईट आहे ही पी साईट आहे, ह्या पी साईडला मूव्ह झालेलं दिसतं. ओके, that always remember, this is the most important statement or the most important thing. It is seen during the elongation, that is the dipeptide it is initially present at the A site. During the translocation they move from A site to P site. म्हणजे इथे A site काय झाली vacant झाली. So because of that the A site become vacant. So this is the vacant A site. म्हणजे ए साईट रिकामी होते. ज्यावेळेस ए साईट रिकामी होते त्यावेळेस हा जो पी साईटला हा जो टी आर एन ए आहे की ज्या टी आर एन एने जो अमिनो ॲसिड आहे तो अमिनो ॲसिड हा सेकंड अमिनो ॲसिडला तिथे कनेक्ट केलेला आहे विथ द हेल्प ऑफ पेप्टाइड बॉन्ड. आणि मग हा टी आर एन ए जो आहे तो एम्प्टी राहतो किंवा त्याला आपण अनचार्ज्ड टी आर एन ए असं म्हणतो. सो दिस इज द अनचार्ज्ड tRNA, and that uncharged tRNA it is kicked off from the P site, and that uncharged tRNA they release outside from E side. So the name is given exit site. हा जो tRNA आहे तो ही जी E site आहे ह्या E site मधून हा इथे exit घेतो and they float back in the cell cytosol. Okay, and then what happens student, then here again the A site it become vacant. So the A site it become vacant, ती रिकामी राहते. Then for what purpose they become vacant, they receive the further amino acyl tRNA complex. Okay, same as like this पूर्वीप्रमाणे. लक्षात येतं. म्हणजे इथे पुन्हा अमिनो ॲसिड टीआरएनए कॉम्प्लेक्स, इथं थर्ड अमिनो ॲसिड टीआरएनए कॉम्प्लेक्स इथं रिसीव होईल. बरोबर आहे. आणि इथे अमिनो ॲसिड कोणतं असेल, दिस इज द अमिनो ॲसिड AA3. ओके, AA3. अँड देन आफ्टर दॅट इन द प्रेझन्स ऑफ रायबोझाईम एन्झाईम, ओके, रायबोझाईम एन्झाईम अगेन the peptide bond it is developed. Okay, peptide bond it is developed in between the amino acid A2 and A3. Now आता इथे काय तयार झालेलं आहे की ट्रायपेप्टाइड स्ट्रक्चर इट इज डेव्हलप ॲट ए साईट. ओके, ट्रायपेप्टाइड स्ट्रक्चर इट इज डेव्हलप ॲट ए साईट. आणि पुढे काय होणार आहे मग, दिस ट्रायपेप्टाइड स्ट्रक्चर दे मूव्ह फ्रॉम ए साईट टू पी साईट. आणि नंतर कोणती साईट एम्प्टी राहणार आहे, व्हॅकंट राहणार आहे, ए साईट ही व्हॅकंट राहील. ओके, सो यू हॅव टू रिमेंबर. देन पुढे बघा, पुढच्या स्टेपला हा रायबोसोम पुन्हा पुढे सरकेल. परंतु पुढे सरकल्यानंतर रायबोसोम काय करेल, हे पुढचे दोन कोडॉन ॲक्वायर करेल. पण शेवटचा कोडॉन कुठला आहे, UAA हा टर्मिनेशन कोडॉन आहे. मग टर्मिनेशन कोडॉन किंवा आपण त्याला स्टॉप कोडॉन म्हणतो, तो स्टॉप कोडॉन हा कोणतंही अमिनो ॲसिड अट्रॅक्ट करत नाही. दे नेवर रेकग्नाइज एनी टाईप ऑफ अमिनो ॲसिड. मग इथे थर्ड स्टेपमध्ये आपल्याला इथे दिसतं की हे दोन कोडॉन म्हणजे CCA आणि UAA हे दोन कोडॉन हे ह्या रायबोसोमने पुढे जाऊन ॲक्वायर केलेले दिसतात आणि त्याच्या अगोदरचे कोडॉन म्हणजे AUG आणि GCG हे दोन कोडॉन AUG आणि GCG हे दोन कोडॉन 5 प्राईम एंडला एक्सपोज झालेले आहेत. बरोबर आहे. म्हणजे इथे पुन्हा लक्षात घ्या की AUGमुळं AA1 एक अमिनो ॲसिड ॲड झालं, दुसऱ्या कोडॉनमुळं दुसरं अमिनो ॲसिड ॲड झालं, तिसऱ्या कोडॉनमुळं हे तिसरं अमिनो ॲसिड ॲड झालं, पण थर्ड लास्ट कोडॉन इट इज the nonsense codon or it is a stop codon. Okay, it is stop codon. So that nonsense codon they unable to recognize any amino acid from the 20 different types of amino acid. So toward this nonsense codon there is no any type of the amino acid tRNA complex received. So here release factor they attach at the nonsense codon or at the stop codon. Release factor they are attached, and so when the release factor they are attached, then this elongation of polypeptide chain it is immediately stopped. Okay, elongation of polypeptide chain it is immediately stopped. म्हणजे तुमच्या लक्षात येत असेल की mRNA वरती हे 4 कोडॉन आहेत, 1 2 3 4 आणि शेवटचा कोणता आहे, हा नॉनसेन्स कोडॉन आहे किंवा स्टॉप कोडॉन आहे किंवा आपण त्याला टर्मिनेशन कोडॉन म्हणतो की जो कुठलंच अमिनो ॲसिड अट्रॅक्ट करत नाही किंवा या स्टॉप कोडॉनकडे कुठलंही अमिनो ॲसिड tRNA कॉम्प्लेक्स अट्रॅक्ट होत नाही. म्हणजे या अशा mRNA पासून जे पॉलिपेप्टाइड चेन फॉर्म होईल किंवा जे प्रोटीन फॉर्म होईल त्या प्रोटीनमध्ये किती अमिनो ॲसिड असणार आहेत, फक्त 3. लक्षात घ्या, ज्या mRNA वरती लक्षात घ्या ज्या mRNA वरती 4 कोडॉन आहेत, 4 कोडॉन आहेत, त्या mRNA पासून जे प्रोटीन तयार होईल त्या प्रोटीनमध्ये फक्त 3च अमिनो ॲसिड असतील, हे लक्षात घ्या. समजा त्या mRNA वरती 100 कोडॉन असतील, 100 कोडॉन्स असतील, तर तयार होणाऱ्या पॉलिपेप्टाइड चेनमध्ये किती अमिनो ॲसिड्स असतील, किती असतील, 100 मधला एक हा स्टॉप कोडॉन असेल किंवा नॉनसेन्स असेल आणि बाकीचे 99 हे सेन्स कोडॉन असतात. म्हणजे पॉलिपेप्टाइड चेनमध्ये किती अमिनो ॲसिड्स असतील, 99, हे लक्षात घ्या. सो यू हॅव टू रिमेंबर दिस. अँड व्हेन देर इज ए लास्ट कोडॉन, the stop codon or nonsense codon, and towards that nonsense codon the release factor they get attached there, the elongation of polypeptide chain it is stopped. I think you understand well. The second step of the translation, that is the elongation of polypeptide chain. Then the last step of translation it is the termination and release of polypeptide chain. Termination and release of polypeptide chain. Remember when that release factor they are attached at the termination codon, then that elongation they immediately stop there, and it is the time where the termination is there and the termination of polypeptide chain will be occur. So remember here, in the presence of the magnesium ion at a suitable concentration, these two subunits they associate. But remember, when that release factor they attach at the stop codon, then the concentration of this magnesium ion it is decreases, and when it is decreases, then these two subunits means larger and smaller subunit they dislocate from its position. म्हणजे हा larger subunit इथून निघून जातो, smaller subunit इथून निघून जातो. नंतर हा जो last tRNA आहे, हा लास्ट tRNA पुन्हा डिस्लोकेट होतो आणि तो सेल सायटोप्लाझममध्ये निघून जातो. दे फ्लोट बॅक इन द सेल सायटोसोल. देन दॅट रिलीज फॅक्टर दे डिस्लोकेट फ्रॉम द लास्ट कोडॉन अँड द mRNA it is also released in the cell cytoplasm. But remember here, as compared to the lifespan of the other RNAs, the mRNA they are having the very short lifespan. After protein synthesis, finally the mRNA they get denatured. Okay, they get denatured because it will be immediately digested by or denatured by the nucleases enzyme. Okay, nucleases enzyme, and so because of that, that mRNA it is short-lived. So, student, here we have completed the entire process of protein synthesis. Okay, in the process of protein synthesis, uh, we have seen there the two main processes of the protein synthesis, that is the first process it is transcription process, and the second main process it is the translation process.
Steps of the translation process. Initiation. The small ribosomal subunit binds to the initiator tRNA carrying the initiator amino acid methionine. This complex then attaches to the cap structure at the 5' end of an mRNA and scans for the start codon AUG. The process is mediated by several initiation factors. At the start codon, the large ribosomal subunit joins the complex and all initiation factors are released. The ribosome has 3 sites. The A site is the entry site for new tRNA charged with amino acid or aminoacyl tRNA. The P site is occupied by peptidyl tRNA, the tRNA that carries the growing polypeptide chain. The E site is the exit site for the tRNA after it is done delivering the amino acid. The initiator tRNA is positioned in the P site. Elongation. A new tRNA carrying an amino acid enters the A site of the ribosome. On the ribosome, the anticodon of the incoming tRNA is matched against the mRNA codon positioned in the A site. During this proofreading, tRNA with incorrect anticodons are rejected and replaced by new tRNA that are again checked. When the right aminoacyl tRNA enters the A site, a peptide bond is made between the two now adjacent amino acids. As the peptide bond is formed, the tRNA in the P site releases the amino acids onto the tRNA in the A site and becomes empty. At the same time, the ribosome moves one triplet forward on the mRNA. As a result, the empty tRNA is now in the E site and the peptidyl tRNA is in the P site. The A site is now unoccupied and is ready to accept a new tRNA. The cycle is repeated for each codon on the mRNA. Termination. Termination happens when one of the 3 stop codons is positioned in the A site. No tRNA can fit in the A site at that point as there are no tRNA that match that sequence. Instead, these codons are recognized by a protein, a release factor. Binding of the release factor catalyzes the cleavage of the bond between the polypeptide and the tRNA. The polypeptide is released from the ribosome. The ribosome is dissociated into subunits and is ready for a new round of translation.
So student, I think you understand well what is meant by the protein synthesis and how that protein it is synthesized intracellularly, in the sense inside the cell cytoplasm, and how DNA they control over the entire process of the protein synthesis. So all things I think you understand well. So student, with this we will stop at this moment.